What's your tonicity, or is it your osmolarity? You can't learn by osmosis, but you can certainly learn about osmosis, and if you learn about osmosis, you'll realize that learning by osmosis, that phrase doesn't even really make that much sense, because osmosis is referring to the movement of solvent, and I would think that, like, ideas and knowledge and stuff, that would be more like solute, like dissolve things, but anyway, let's step back and discuss what the heck I'm talking about.
To avoid any confusion, let's start with talking about diffusion. Then we'll get into osmosis and osmolarity and tonicity and how osmolarity and tonicity are different. And we'll look at some places that this shows up, such as in biology and medicine, and in the lab. So let's start with diffusion. So diffusion, we're probably all familiar with it, even if we don't know the terminology. If you were to stick a drop of dye into some water, the dye is originally very concentrated, and then it's going to spread out throughout the water until you have an even distribution of the dye. So in more molecular terms, what's happening is that at the net, that these molecules, so like the dye molecules, so your dye is an example of a solute, it's a dissolved thing, it's dissolved in the solvent, so in that case, the water. And the solute is it's moving around randomly, and so is the water, but it's going to move around randomly in such a way that at the end of the day, or once you reach a dynamic equilibrium, the molecules are still moving around, but they're evenly distributed throughout. So if you have a lot of molecules in one place and they're all moving around randomly, then they're going to move around randomly, and a lot of them are going to move this way, say, a lot of them are going to move this way and this way and this way and this way. And because they're moving randomly, ultimately they're going to be evenly distributed throughout. If you didn't have you can imagine why it's going from like a high concentration to a low concentration at this like net level. If you have something at a low concentration, it's not going to move. It's less likely that since you have fewer molecules over here, it's less likely that they're randomly going to move over here than it is that if you have a lot of molecules over here, more likely that some of those are going to randomly move over there to where there's fewer of them. And so at the end of the day, once you reach this dynamic equilibrium, the molecules are still moving randomly around, but because they're evenly distributed, their random movings are going to cancel out. And so you have this equal distribution. And you can say like on the net effect is that you have this movement from a high concentration to a low concentration. So here we're talking in terms of the solute concentration, but we can also talk in terms of the solvent. And when we talk in terms of the solvent, then we're talking about osmosis. So with osmosis, we're talking about the movement of the solvent, not the solute. So it's still diffusion, but it's diffusion in terms of this. We're talking about the molecule that's diffusing is going to be the solvent, whereas typically, like in a case like this, we were thinking of the movement being the solute. So if instead of just free diffusion, we have a membrane there or some sort of partition, if this partition doesn't let anything through, say it's like a lead wall, then we'll say that it's fully impermeable. If it lets some things through, if it lets everything through, we'll say it's fully permeable. And if it lets some things through but not others, we say it's semi-permeable. And so if it is fully permeable, everything would diffuse equally throughout. You get this even distribution on both sides. And so when you reach that dynamic equilibrium, you're going to have an equal concentration of everything spread throughout. If it's fully impermeable, nothing's going to happen. But what if it's somewhere in between? If it's semi-permeable, so it's permeable to some things, but not others. So if the things that it's permeable to, we say those things are penetrating, and the things that it's impermeable to, we say they're non-penetrating. In the case of osmosis, osmosis is when we're talking about diffusion of our solvent, so commonly water. And in the strictest definition of osmosis, we're considering that all solute is impermeable. So we consider that no solute can go through and that the water can. And so what's going to happen is that the water is going to be at a lower, like free concentration of water when there's a bunch of solute around, and a higher free concentration of water when there's not. So there's more water molecules over here, the molecules can move from either side, but since you have more over here, they're more likely that you're going to have water molecules move from this side to this side. And when you reach equilibrium, you're going to have this distribution where you have more of the water over here. You're going to have this net movement of water over here to the side that had more of the solute. But membranes are not always strictly permeable to solvents, but nothing else. So you can have membranes that are permeable to some things, but not others. And we see this in dialysis. So I have a post on this the other day where basically you stick your protein in or whatever in a patch, in a little pouch, and then you can desalt it because your protein is stuck in this pouch, but these little things like the salts can flow through. And so the membrane is going to be permeable with respect to the solvent and the salts, but impermeable with respect to the protein. And so in this case, our membrane is not just that simple case of when we had this membrane that only lets through our solvent. And so a lot of times in biology and biochemistry, we're dealing with situations that aren't as clear cut as this like classic osmosis example. And when this comes in, this can get kind of confusing when we're talking about things like osmotic pressure. And instead, we can use terms dealing with tonicity, which are more dealing with the effective osmotic pressures and osmolarities and stuff. So tonicity is when we actually take into account whether with tonicity, we actually take into account whether the membrane is permeable or not. And therefore, we only basically consider the effects of the non-penetrating solutes. So the things that can't move, because if the things can move, then they can kind of like even themselves out and they don't need the water to pull all the weight in evening out the solutions. So keep in mind that when I'm going to introduce these topics of osmotic pressure and osmolarity, when we're dealing with semi-permeable membranes that are semi-permeable with respect to the solutes as well, so they let some solutes through but not others, they're more restrictive. In that case, the same concepts are going to apply in that we're going to have this pressure that's generated by the dissolved molecules. But here we're only going to consider the dissolved molecules that are penetrating. And so keep this in mind. So with osmosis, this, however, is we're looking at the solution level and not taking into account the membrane. With tonicity, we take into account the membrane and whether or not the particles can act as like, you can sometimes see this term like osmotically active, so that these particles are non-penetrating, they can affect osmosis, the osmotic pressure, they can affect the osmosis, whereas those that aren't penetrating the ones that are penetrating, they're not going to matter because they're going to be able to kind of like even themselves out. So keep this in mind. We'll get back to tonicity in a minute. But let's deal with this idea of osmotic pressure. So osmotic pressure is kind of, it explains this mystery you might be thinking. So if you have a bunch of solute molecules over here, and they can't come over here, you can move water over here to kind of like dilute this out, but you're never going to have equal concentrations on either side. And remember that when we talk about that like ideal, we think of the like end goal of diffusion or whatever is to like have equal concentrations, but we can never have that unless we take all of the water out of this side. But this isn't what's going to happen. When you reach this dynamic equilibrium, you're going to have more of this water that's gone through, but you're not going to be dry. You're not going to have all of the water go through. And this is because there's a counterbalancing force, this osmotic pressure. So basically, if you imagine a U-shaped tube and you have a solute on one side that is non-penetrating, so it can't get through, so it is osmotically active, it is going to affect things. And then you have water throughout. What's going to happen is that when you reach equilibrium, to reach equilibrium, the water is going to flow through here, but it's going to stop when it gets to this point where basically the added water, as you're pushing the water up here, it's going to generate this hydrostatic pressure. This pressure is going to be pushing down on this water and making it harder for the water to move up the tube. This pressure we can describe as osmotic pressure, and it's the hydrostatic pressure generated by the added water when osmosis stops on its own. So if you don't do anything, what if you were to do something? If you apply that, this is telling us too that this is the amount of pressure we would need to apply in order to prevent the osmosis from happening in the first place. So in order, we could prevent this movement of water if we applied that much pressure. If we apply even more pressure, we can actually force the water back through the other way. And this is the idea with the reverse osmosis, which is used to purify water by applying more pressure than is the osmotic pressure. This osmotic pressure is a colligative property. What this means is that it depends only on the amount of the dissolved particles and the temperature, and not the identity of the particles. So we talked about colligative properties before when we were talking about like boiling point elevation and freezing point depression. And a key thing with colligative properties is that it depends on the number of dissolved particles and not the number of like things that you put in, if that makes sense. So basically this comes into play when we're dealing with electrolytes. So things that dissolve and dissociate. So if you take a molecule of sucrose and you stick it in water, it can dissolve in that, meaning that each of these sucrose molecules is going to get a coat of water. But you see that this glucose and the fructose that make up sucrose are going to stay together. So you get the same number of molecules that you put in, or you get the same number of like things that you put in. What about a salt? So like if you take sodium chloride, now when you stick this into water, the electrolyte is going to dissolve and dissociate. So you put in one formula unit's worth of sodium chloride, and you get a unit's worth of sodium and a unit's worth of chloride. So now you have 2 units worth of the thing that you put in. So you have like twice as many particles as like units that you put in. And so we can actually use this, there's this like Van 't Hoff factor, which takes into account how many particles this unit breaks into when we're determining molarity of dissolved particles. And we have this term called osmolarity, which we use when we're applying, when we're talking about dissolved particles. So if you apply that Van 't Hoff factor and stuff, and you figure out the number of dissolved particles, you can figure out the osmolarity. And so if you put that, if you start with 1 molar of sucrose, so a mole is just a 6 times 10 to the 23rd or something, like a Dunton, but a lot bigger because we're dealing with really, really tiny molecules. And so really, really big quantities of those really, really tiny molecules. And so we're typically dealing with micromolar, nanomolar, millimolar, those sorts of units in a biochemistry lab. And if we're talking about molarity, this can be of anything. So this is why I can talk about like molarity of chocolate chips and stuff. But when we're talking about osmolarity here, we're talking about dissolved particles. Instead of a tube of Rolos, if the Rolos then dissociated, so they came apart into the individual Rolo candies, that would be like my osmolarity. And so osmolarity is helpful when we're trying to figure out things like osmotic pressure, which are colligative properties. And we can compare osmolarities with terms. So we have hyperosmotic, meaning that there's more dissolved particles. So hyper over isoosmotic, which means that they have the same amount of dissolved particles. I so don't care where I go because everywhere is the same. Or hypoosmotic, which means that they have fewer dissolved particles, hypo below. And so we can say that so if you took a 1 molar sucrose solution and a 1 molar sodium chloride solution and we dissolve them, the sucrose solution would be hypoosmotic to the sodium chloride solution, which would be hyperosmotic to it. And so we can use these terms. And if we're talking about determining osmotic pressure, then this is going to allow us to compare. So we can say, okay, if the osmolality of one side had a higher osmolality, then water is going to flow towards that side. But this is, remember, in the strictest sense, when we're just comparing these osmolalities, the osmotic pressure is going to be a property of that solution without taking into account the membrane. So you can see that in our example, when we're comparing sodium chloride and sucrose, we weren't even considering whether or not those molecules themselves could actually get through the barrier. And if some of those molecules can't get through the barrier, then we want to take that into account when we're trying to do practical applications of things, like thinking about how molecules are going into cells and how our protein is going to act in that dialysis pouch. And so, we can take into consideration the membrane and the relative permeability of the solutes. So we need to take into account like the membrane and the solute and how they interact, and in order to actually predict where water would flow in such a situation. And so this concept of tonicity is going to take this into account. So tonicity is going to take into account like the osmotically active particles. So it's going to take into account things that are non-penetrating, things that can't get through the membrane, and thus things that are going to actually contribute to this osmotic, to the actual like effective osmotic pressures that you're going to see, and to the effective osmolarities, so that you're going to basically, you'll have the water move from the side that is hypotonic, so the side that has less of the solute, more of the water or the solvent, a higher like free concentration of the solvent. So it's going to flow from its high concentration to its low effective concentration. So it's going to flow towards the side with more solute, ultimately kind of diluting things out. And then you get this osmotic pressure that is stopping it from going all the way. So tonicity, it's not a value, like we had a value, like we could calculate an osmotic pressure, we could calculate a molality. Tonicity isn't such a value, it's like a comparative term. And so we can say something as hypotonic, hypertonic, or isotonic compared to some other solution, but you can't say a solution has a tonicity of X like you would with osmolarity or osmolarity. But we are able to compare these. And the other thing is that with tonicity, in addition to comparing these solutions, it's comparing these solutions in the context of this particular membrane. And so you have to take into account the membrane and the solutes and the relative permeability of them. And that way you can then compare where the solvent is actually going to flow. So if you have a hypertonic solution, you're going to have more non-penetrating particles. So the side with more of the solute is going to be hypertonic to this side, which has fewer non-penetrating particles, and therefore we call this side hypotonic. And at the end, we're going to have isotonic, which means that they have the same amount of non-penetrating particles, except that we can't actually get there because of the pressure. Because this is talking about non-penetrating solutes, not all solutes, the tonicity and the osmolarity can sometimes be like not consistent in terms of which prefix. Like you can't say that. So isoosmotic solutions are not always isotonic. Hyperosmotic solutions are not always hypertonic, but hypoosmotic solutions are always hypoosmotic solutions are always hypotonic. And so let's look at some examples of how this can be. So say we have this solution where we have this barrier, but we start with a solute in only one side of the barrier. But this solute is penetrating. So we have these two solutions. And these solutions are this solution is hyperosmotic because it has more dissolved things. This side is hypoosmotic because it has fewer dissolved things. But what's going to happen is that they're actually isotonic because the solute is penetrating. So they have the equal amounts of non-penetrating solute because all of the solute is penetrating. And so since the solute is moving, you're not going to have that net movement of water because you're going to have the movement of the solute and the water. And so you're going to get this even distribution, and you don't have any of this like tone imbalance. What happens if you have non-penetrating solute, however? Here, if all your solute is going to be non-penetrating, now the solution is going to be hypertonic because you have a higher concentration of non-penetrating. It's going to be hyperosmotic because you have a higher concentration of dissolved things. And then this side, conversely, is going to be hypotonic and hypoosmotic. And so what's going to happen now is your solvent is going to move. You are going to have this net movement of the solvent. So this, you're going, because this was hypertonic, this was hypotonic, now you have the movement of the solvent from the hypotonic to the hypertonic, and you're going to, now you end up with kind of like diluting out the solute. And notice that we're not running dry because this is going to be creating osmotic pressure that's going to be preventing this, that's going to be kind of like stopping the flow eventually. Now our final example, what if we have this non-penetrating and penetrating solute? So if we start with this equal amounts on like both sides, so in that case, since you have equal amounts of dissolved things on both sides, this would be isosmotic. But this side would be hypertonic because if this solute cannot penetrate, but this one can, then this solute side is going to be hypertonic because it has more non-penetrating solute than this side, and this side is hypotonic. And tonicity, remember, this depends on, the tonicity is going to be the thing that predicts where the solvent is going to flow. And so the solvent is going to flow through this membrane towards the hypertonic side. So this might seem like some abstract concept. So let's look at some practical examples of where this comes into play. So one place is like with medicine, with like IV fluids. So your body, as we'll talk about, you have a lot of mechanisms in order to try to keep consistent osmolarity and tonicity inside and outside of your cells. Now imagine that you put an IV solution into somebody. There, if we consider these two common IV solutions, normal saline, which is 0.9% sodium chloride, and D5W, so 5% dextrose or glucose in water. So these are going to be isoosmotic to each other and to cells. So they have an equal amount of dissolved stuff. But the normal saline is isotonic to cells because the sodium chloride doesn't enter the cells. So basically what you put in is, osmolarity wise, is also what you put in tonicity wise in a way. So basically what things are staying out and things are staying in, and you're talking about the movement of the solvent, which in this case isn't going to move on net because you have equal concentrations of non-penetrating things inside and outside of the cells. But D5W is hypotonic because the glucose can enter the cells, so it's penetrating and it doesn't like count when we're determining tonicity. So what's going to happen is if the glucose enters the cells, now you have a your solution is going to be like more dilute outside of the cells and inside. And so this is going to basically lower your osmolarity outside, even though you started with osmolar solutions. And so this is going to make the solution hypotonic. And so water is going to flow into the cells. You can think of it kind of like helping dilute out the cells, or think of it as kind of the, because you have more water particles outside of the cells and water can move into the cells, they're more likely to move into the cells than they are to move out of the cells. And so this is, doctors can also use different concentrations of solutions if they want to try to alter the balance of fluids in a person's cells or extracellular fluid. And so this is just a good example that isosmotic solutions are not always isotonic, hyperosmotic solutions are not always hypertonic, but hypoosmotic solutions are always hypotonic, because if you have less than something, it doesn't matter if that something can go in or not, it's always going to be less. So some other lessons to take away from this, even if you're like stranded at shipwrecked, you don't want to drink salt water. One, because it's gross and there's like gross stuff in there. And two, because it's hypertonic. So there's a lot of salt in there, more salt than there's stuff in your cells. So it's going to pull water out of your cells and dehydrate you. But even if you are dehydrated, you don't want to give a pure water IV because that's going to be hypotonic and that's going to burst your cells. So this brings some important concepts about basically if something is hypotonic, then water is going to flow in, and this can cause things to burst. If things are hypertonic, water can flow out, and this can cause things to shrivel up. So in biology, we see a lot of mechanisms to try to compensate for this. So in our blood, we have this really abundant protein called serum albumin, and it's going to help maintain the osmotic balance so water doesn't flow into cells. So you have a lot of stuff going on in your cells. You have a lot of stuff in your cells. There's a lot, lots and lots of molecules. And so you don't need as many different things outside, like in your blood, as you do like going on in your cells. But you need to have stuff in your blood that's going to prevent the water to prevent your blood from being hypotonic, which would make all the liquid, like all the water, flow into your cells and then make all those cells burst. So serum albumin helps maintain this osmotic balance so that the water doesn't flood into cells. Plants and bacteria, so they actually take advantage of osmotic pressure. So they have, unlike our cells, they have strong cell walls. So we don't have strong cell walls, we just have cell membranes, but they also have cell walls. And these cell walls are going to let them resist osmotic pressure. So what plants do is that they have these, but what happens is they have these like vacuoles and stuff, and water that enters pushes against the cell wall. And this is like it's like fruitlessly trying to expand it. And this is going to create hydrostatic pressure that provides a crisp stiffness, which makes for crispy vegetables. Another osmotic trick, cells, because remember they have a bunch of stuff already, they're going to store their energy in the form of polysaccharides, so like starch and glycogen, which are large chains of simple sugars, instead of lots of simple sugars. So instead of lots of individual glucose, if you link those up, you have fewer dissolved particles, and so they can store the same amount of energy with lower osmolarity. There's also some random or general places that osmolarity shows up. So you might have played with one of those like magic row toys. These have super absorbent polymers, and we'll talk more about a concept like this when we talk about transfection reagents and like PEI and cationic carriers. But these have a lot of positively charged groups that are going to pull in water, and then it's going to grow, but it's going to be counterbalanced by that osmotic pressure. And so your snowman isn't going to grow to the size of your house or anything. Also, don't let your dog pee on grass. Their pee is going to be hypertonic. So it's going to pull water out of the grass, and this is going to lead to a dead patch. So now let's look at some places where osmolarity shows up in the lab. A common method in the lab is, if you're in like a molecular biology or biochemistry lab, is an agarose gel electrophoresis. We have this gel that's made out of this long sugar chain agarose, and it makes this gel that we can then send DNA fragments traveling through to separate them by size. One thing you might have noticed if you were to leave one of these gels be is you go away and then there's a pool of liquid on top of the gel and your gel's a little shriveled up. You also might have seen this on like a cheesecake or something like that. What's happening is this thing called syneresis. So in your gel, you have this mesh that is filled with water. And because there's going to be like more water inside of the gel than outside of the gel, you're going to have this osmotic pressure from the water kind of like trying to escape, but it's trapped in this gel. And it's going to start swelling, but it's going to be counterbalanced by this elastic pressure from the gel walls wanting to contract. So if the osmotic pressure isn't strong enough, then what can happen is that the elastic pressure can win, and you can kind of get this collapsing and this rearrangement that kicks out water. If you can increase the osmotic pressure, you can prevent that. So one way to do this is by adding charged groups to the agar so that it is going to pull in more water and prevent that water from leaving. And in this way, you have increased osmotic pressure and you prevent this syneresis and this like escape of the liquid. And so this is often used as like when in cooking with thickening agents to increase the charge and various properties in order to prevent this syneresis from happening, although we wouldn't want to introduce charged things when we're doing an agarose gel because that's going to interfere with the movement of the DNA. Another place where we see osmotic pressure come into play is when we're doing a mini prep, which is a way that we can isolate plasmid DNA, so these circular pieces of DNA, often that we've cloned some gene that we want, like a protein expressed from or something. We put in the instructions for making that protein into the circular piece of DNA called a plasmid, and we stick that into bacteria, and the bacteria make a lot of copies of it. Using this alkaline lysis, this like mini prep kit, we can then isolate out that plasmid and do things with it. In order to isolate that plasmid, we're going to have to, in order to isolate that plasmid, one of the steps in order to isolate that plasmid, we're going to have to break open those cells. But before we break open those cells, we want to make sure that we're breaking them open into an environment that we want them in. So we're going to resuspend them. We're going to pellet them, so centrifuge them to spin down the cells, remove the media, so the food they were growing in, and put it in this other buffer. And when we put it in this other buffer, this resuspension buffer, we want to make sure that it's not going to be lysed already. We don't want to break it open yet. And so one way to prevent this from breaking open is you add glycerol. This glycerol is going to help maintain the osmotic balance. So you can imagine if you had too much water, if you had like a really dilute solution, then what's going to happen is that that's going to be hypotonic. Water is going to move from the solution into the cells, and the cells are going to swell and swell and swell and burst. And we don't want that to happen, so you can add glycerol, which is going to maintain the osmotic balance, adding things outside so that the water, you have a higher concentration of these non-penetrating things outside. This is going to make this isotonic so that the cells aren't going to break open. Another place we need to consider osmotic balance is when working with bacteria is when we're actually just like feeding them their media. So a common bacteria food is lysogeny broth, LB, and it has salt, which is going to do, among other things, it's going to help keep the osmotic balance. So by having salts outside, you're going to once again prevent too much water from going into the cells, increase the tonicity outside. Finally, we had talked about those like magic grow snowmen. And I mentioned that a similar concept is used when we're talking about transfection with cationic carriers, such as positively charged polymers, such as PEI. These have a bunch of different ways that they work, such as because they have a positive charge, they can kind of balance out hide the negative charge of the DNA and allow it to get into these cells, which have this negative charge in their membrane. Often what happens is that these are taken into these endosomes, so these like pouches inside of the cells. And when they're taken into these pouches, what's going to happen is that they're going to take water with them. So this is not from this, but you can imagine that you have the water coming in. When the water is coming into this pouch, then that's going to make this endosome pouch kind of lyse. But this is happening inside of your cell, and this is going to allow the DNA to escape inside of your cell. And so here again, we have osmotic pressures coming into play. So to review, osmosis is the movement of water or another solvent through a semi-permeable membrane in this random manner that ends up with the effect of basically balancing out the overall concentrations of stuff on either side. And we're talking about stuff here, we're talking about solute, so dissolved things. And osmolarity is the amount of dissolved solute particles of any kind, so the composition doesn't matter. Diffusion is a more generic term for the movement of stuff or the solvent. And so with osmosis, we're talking about the solvent. And when we're talking about diffusion, we can also be considering the movement of solute. Osmolarity is a measurement of the concentration of all the stuff, so we don't care, molecules, any kind, composition, it doesn't matter. Whereas tonicity is a comparative term that only takes into consideration the concentration of non-permeable stuff, so stuff that can't get through the membrane on either side of a non-permeable barrier, and tells you in which direction you'll have the net movement of the solvent. So we only care about the non-permeable stuff because the permeable molecules can basically balance themselves out. So they'll equally increase osmolarity on both sides of the barrier. But the non-permeable molecules will only increase osmolarity on the side that they're on. And so water has to pull the weight. It has to be moving in order to balance out those osmolarities. In a hypertonic solution, you have higher concentrations of non-penetrating solvents. So hyper, over, and so the water is going to move out. This is why if you were to stick a cell into a high salt solution, the cell would shrivel up. Hypotonic is lower concentrations of non-penetrating solvent. So hypo would be low, and here water is going to move in. So if you were to stick a cell into pure water, this water would flow in and the cell would burst. And so I hope that this helps you understand some of these concepts of osmolarity, tonicity, and these kind of confusing terms, and why it really does matter, and how it comes into play, and why molecules are just really cool.